Did you know that the zebrafish, Daniel Kang, originates in the Ganges River in India, and also freshwater bodies surrounding that country? This particular organism has become an invasive species in Alabama. Now, what is the importance of this zebrafish? Well, you see, the zebrafish contains what you would call heat shock proteins, and these heat shock proteins are able to regulate cellular regeneration. Therefore, in extreme conditions, these zebrafish are able to reproduce certain proteins of their body again and again and cope with dangerous mechanisms. Now, what application does this have to medicine? Well, you see, people with diseases that have muscular dystrophy or just painful diseases like cancer can apply these heat shock proteins and maybe hopefully find a way to regenerate lost tissue and possibly organs. But there's so many heat shock proteins. In fact, there are too many that we have to study. But one in particular that we want to study is heat shock protein 70. Now, where does the number 70 originate from? Well, you see, proteins are measured by their molecular weight in kilodaltons. Therefore, heat shock protein 70 is measured 70 kilodaltons. Now, how do we measure these proteins? Well, obviously you can't use a ruler. That's not a way to measure a protein. Special devices have to be used, such as an electrophoresis gel and a Western blotting mechanism. Now, to process these tissues, particularly the proteins, the muscle cells of the zebrafish have to be collected. Now, since these fish are small and very capable of being caught, since they are so invasive in Alabama freshwater systems, we have easy access to them. Thankfully, the wonderful school of JSU, Jacksonville State University, has obtained several zebrafish over the past years. So we are in a high quantity of these organisms. First, we will collect a sample of zebrafish, roughly about 30 of them. They will be in 3 experiments. 1 is in high temperature, 2 is in low temperature, and 3 is the control, which is just at room temperature. Now, since these zebrafish are so hardy, they can survive all 3 temperatures. But what we want to see is how their heat shock proteins, particularly heat shock protein 70, react to these external factors. So come on, let's discuss how this process is taken. Muscle tissues from the gills and white skeletal muscles will be taken from the fish and homogenized. Homogenized means just grounded up, mixed up together, so that the tissues and proteins are easily processed. These particular tissues will be undergone in a centrifuge treatment, which is just basically a rapid rotation of the tissues so that the proteins are able to separate from the actual muscles. After roughly about 3 to 4 centrifugations, these tissues will undergo what you would call a SDS page on a 1-dimensional scale and 2-dimensional scale. Now, what are the differences between these dimensions? 1-dimensional SDS page requires just a particular gel sheet. 2-dimensional SDS page requires 2 gel sheets. So there is going to be further separation between each sheet. Once the proteins are loaded on the sheet, you'll be able to see how far these proteins travel and to what extent they travel. That is how we determine the molecular weight of each protein. From that point, we can determine which protein is heat shock protein 70. Once determined, we will perform a Western blot procedure. This will actually separate the protein onto another sheet of gel, and therefore we can isolate it and further study the protein. Now, none of this is possible without help. And what I mean by that is we need funds to provide for our research. These machines that we're using are by no means cheap. So we require monetary assistance for this. The fate of the world is at stake. We want to cure cancer. We want to cure disease for our generations to come. Please, help.